Great. So first, our first contributor is Xiao, who will talk about not only what is in the program, but something else as well.
Hi, everyone. I'm going to talk about genetic and RNA variants in RNA-seq and the methodologies that our lab developed over the past few years. And because this is the first talk, so I will give a very brief introduction of RNA-seq, and I'm sure that most of you do not need this introduction, so it will be quick. So first of all, in RNA-seq, we isolate RNA and then make libraries using different methods, and then sequence the fragment ends. You can do paired end or you can do single end reads. And then after you get the reads, then you can map to the genome or transcriptome or both. And then some reads map to the exons, some reads map to exon-exon junctions. So that will allow us to do different types of analysis. So obviously, the different types of analysis, the first most often used is gene expression analysis. And there are a number of tools developed for this. And then there are isoform analysis or alternative splicing analysis, and there are many tools developed for this purpose as well. And in fact, today we have a number of exciting talks lined up to address these questions. So I'm going to talk about single nucleotide analysis instead. And in this area, there is much less tools, and these tools are just appearing, and some of them are not specifically designed for RNA-seq analysis, but rather for DNA sequencing analysis initially. Okay, so why do we do single nucleotide analysis from RNA-seq reads? And this is just a cartoon showing you the different reads lined up, and there are single nucleotide variants at different positions, and this is typically seen in the RNA-seq data analysis. And other than sequencing errors, which we could detect and we could remove, one type of single nucleotide variants that can be present in the RNA-seq reads would be genetic mutations, obviously. So these are the DNA mutations that are in our genomes, and if they are expressed in the RNA, then we can see the nucleotide variants in the RNA-seq reads. Obviously, detecting these single nucleotide variants that came from the genome would be interesting and beneficial to the genome sequencing projects or to diagnostic purposes. And another type of single nucleotide variants would be RNA editing sites. And this has been in the spotlight in the past couple of years only. And so it's a relatively new area. RNA editing is a post-transcriptional process where after transcription, then enzymes come in to edit the RNA nucleotides. And the most prevalent type of RNA editing is A to I editing or A to G editing, changing an A nucleotide to a G nucleotide in the RNA. And the second type of RNA editing is C to U editing, which I'm showing an example here. So in this ApoB gene, there is CAA encoded in the DNA, and there is a stop codon here at the end. Without C2U editing, then you can generate a longer isoform of the final protein product. But with C2U editing, changing this C to a U nucleotide will create a stop codon so that you will generate a shorter protein isoform. So there are a number of examples of important RNA editing sites in our cells, and they are biologically significant. So we are very interested in analyzing RNA-seq reads and get the genetic mutations and RNA editing sites out of these data. So there are a number of challenges in analyzing single nucleotides in the RNA-seq reads. First of all is read alignment, because our read alignment was typically conducted against the reference genome, which has the reference nucleotides in the DNA. And if in our RNA-seq reads, we have a mixture of the reference nucleotides and a variant, such as the edited nucleotides or a mutation from the genome, then read alignment could bias towards the reference nucleotide because you're using the reference genome. And the second challenge is to distinguish genetic mutations and RNA editing sites, because at the RNA level, they both appear to be RNA variants. Without knowing the genome, then you're not sure whether it was genetic or it was a post-transcriptional modification. So the third and the most significant challenge is to predict the functions of the single nucleotide variants, or SNVs. So if we have a list of genetic variants or genetic mutations and RNA editing sites, do we know what they do? Can we bioinformatically predict their function biologically? And this is obviously a major mission for precision medicine as well. Over the years, my lab has been focusing on single nucleotide analysis to address these challenges. First of all, we developed a new read aligner called RASER, which is a read aligner for SNPs and editing sites in RNA. So it is tailored and specifically designed to optimize the mapping accuracy to single nucleotide variants in the RNA-seq reads. And we also developed methods to identify single nucleotide variants and specifically to distinguish genetic mutations or SNPs from RNA editing sites. And the first method we already published last year, which is called GRAMI, which is genome-independent identification of RNA editing by mutual information. And we're working on a second method, which we call ExpressGV, which the aim is to identify genetic variants. So with the focus on the genetic variants instead of RNA editing sites. And we want to combine these two methods so that we can identify both the genetic variants and RNA editing sites. And lastly, to address the functional challenge, we also developed a series of methods to identify or predict functional genetic variants that may affect alternative splicing, and these are listed here. So in the interest of time, today I will talk about very briefly about GRAMI and about this new method that we are still working on. Okay. So this slide illustrates the basic idea behind GRAMI. And again, the challenge is how do we separate SNPs and RNA editing sites? And this GRAMI method first looks at RNA-seq reads and look at the linkage disequilibrium between different nucleotides. And if we have 2 copies of DNA in mammalian cells, and then we have reads that covering 2 SNPs, and you can see that if the 2 SNPs are heterozygous, they're both heterozygous, then in the RNA, you maintain the haplotype that was encoded in the DNA. So AA always linked to AA and CG linked to CG. And in contrast, if we had a SNP and an RNA editing site in the same reads or same pair of reads, then the RNA editing can disrupt the linkage pattern in the DNA. And you can see that the G, here we are showing A to G editing. So the G nucleotides can occur on either copy of the RNA that was derived from either copy of the DNA. So using this type of information, you can see that if you had a bunch of reads with 2 SNPs versus reads with both a SNP and an RNA editing site, then you can tell them apart. And indeed, that was the case. And this is the test where we calculate the mutual information between a pair of editing sites in the reads and a pair of SNPs in the reads. And you can see that the 2 distributions separate quite well. And so GRAMI uses this idea as the first step to actually segregate editing sites and SNPs, and then it integrates machine learning methods to actually train a model to predict editing sites. So in this new version of this method, where we aim to identify genetic variants very accurately in addition to RNA editing sites, so we start from the reads, and similarly, we calculate mutual information, just as I described, and then we predict which ones are SNPs and which ones are RNA editing sites based on the mutual information, but that obviously cannot give us all the SNPs and RNA editing sites. So what we do is that we use those predicted SNPs and RNA editing sites and do a machine learning methods. Here we use generalized linear models to predict. Okay, so just very quickly, we have pretty good precision and sensitivity for both SNPs and RNA editing sites prediction using some test RNA-seq data where we know the genome information. Okay. And we also conducted experimental validation where we ask whether RNA-seq reads can complement genome sequencing. So in this case, we have genome sequencing to the 30-fold, but this sequencing data set still missed some of the genetic variants, and our method could pick them up. So we could predict some new additional genetic variants that were missed by genome sequencing. And on the other hand, we can be specific in predicting our RNA editing sites. So some of these RNA editing sites are actually annotated SNPs in the database, but in the specific cell line, it was an RNA editing site by prediction. Okay, so I'd like to thank people in my lab who conducted this work. So these people did the computational methods, and Jay and Adele did the experimental validation, and also the funding sources. And because I'm the first talk, so allowed to do this, I guess. Okay, that's all. Thank you.